Hello, everyone. Thank you for having me here today. I really appreciate the opportunity to present my work at Nextflow Virtual Summit 2025. It's great to see you, everyone, and thank you so much for joining me today. My name is Nikhila T Suresh, and I am a postdoctoral researcher in Radcliffe Lab at Purdue University. Today, I will be sharing some practical insights from benchmarking the Nextflow core a single-cell RNA-seq pipeline for mouse prostate single-cell RNA-seq data, focusing on usability, reproducibility, and performance. So here is a quick overview of today's talk. I will start with the problem definition and discuss why preprocessing remains a major bottleneck in single cell RNA-seq analysis. Then I will outline the study objectives, design, and methodology we followed. I will walk you through a decision tree for users, pipeline setup, and comparison of results from different approaches. Finally, I will summarize key takeaways and practical lessons I learned. So let's begin with the core challenge. Converting raw single cell RNA sequencing data into usable count metrics is often a major barrier, especially for new researchers. The process involves multiple steps, including alignment, quantification, quality control, and normalization, each requiring specific tools and parameter tuning. For many labs, especially those without dedicated bioinformatics support, this step becomes the most time consuming and error prone part of the workflow. There are several reasons why processing is also challenging. First, the bioinformatic pipeline themselves are complex, involving multiple dependencies and software environments. Managing these installation and ensuring version compatibility can be frustrating. Second, reproducibility across different computing environments, meaning local machines, clusters, or cloud. It's not always guaranteed. Third, choosing a right alignment or quantification method adds another layer of difficulty. And finally, there is a lack of generalized workflows that can be easily adopted. These issues are particularly pronounced for beginners in computational biology, labs without dedicated bioinformaticians, and researchers who need reproducible results for publication. So, So here are the main objectives behind this study. For this benchmark study, to understand the Nextflow pipelines through the lens of a new user, we use the different objectives. The first one is usability assessment, then method comparison, and practical guidelines. For this, we have used a mouse single-cell RNA-seq data to generate the count metrics corresponding to this data using the enough single cell RNA-seq pipelines available out there. And talking about a little bit about this mouse single cell RNA-seq, it's an already published mouse prostate single cell RNA-seq data, and it is a chronic inflammatory data which has been taken from this mouse prostate so that we can study the impacts of inflammation, and also we can explore the immunotherapeutic responses. So we are making use of this data to generate the count metrics using this particular workflow. And we are also analyzing these three parameters by this study. Then, for this benchmark study, we are analyzing my pipeline through the lens of a new user. The analysis was run on Nextflow version 25.04.6. Using the NF core single cell RNA-seq version for 4.0.0. Our high performance computing environment included GCC 14.2.0 and OpenJDK version 17. This setup represents a typical use case in academic labs working with mouse models and single cell workflows, making it ideal for assessing pipeline usability and reproducibility. The biological sample we used here was a C57 black 6 mouse. This is a particular strain of mouse which we used for this particular study. And it is processed using 10x Genomics NextGem single cell 3 prime kit version 3.1. So it's again, as I already mentioned, it's already published inflammatory mouse prostate model for understanding immune therapeutic responses. So little bit more about this study. We have considered 6 mice, and we have injected pre-activated T cells for getting them inflamed. Then after waiting 10 days, they already got one more boosted dose. Then we have dissociated prostates after 21 days as well as 35 days. Then we did the single cell RNA-seq. Then we also performed histology and flow cytometry down the line. So let's move on to the decision tree, which has been already provided by the Nextflow tutorial to guide users when selecting alignment and quantification tools within NF core single cell RNA-seq. The idea was to help the beginners decide which aligner to use. For example, we have selected Alevin-Fry, CellRanger, and STARSolo. Then we have, because our chemistry was supporting all these but different aligners. Then so then we this is the pipeline we have used. Setting up this pipeline involved configuring NF core single cell RNA-seq through Nextflow. While NF core pipelines are containerized and theoretically portable, we still encountered several configuration and execution issues. But we will get into that later in a couple of slides. So what we did using this setup is we have inputted the FASTQ file using this CSV. We mentioned 2 reads in this sample sheet.csv. Then we have also mentioned the GTF file, and also this is the mouse genome file for which we are using this for alignment. Then we have implemented different aligners, as I already mentioned, CellRanger, Star, and SimpleLeaf. The main functions that are being done by this aligners are listed out here. Then we have generated QC metrics using cell counts and UMI distribution. Then we have also obtained count metrics, H5AD file, and multi-QC report. So once we get these outputs, we are ready for doing the downstream analysis. So while doing this, even though we selected CellRanger, Star, and SimpleLeaf, Star didn't work that way as it's supposed to be, and also we could retrieve the outputs from CellRanger and SimpleLeaf. So while analyzing this output in terms of usability and practical guidance, this particular V3 chemistry, it has been automatically detected by the pipeline, and also there was a good custom configuration support for HPC users. Then the bioinformatics AI agent Secura was also it was so helpful for me. And there was additionally there was a community curated pipeline support as well. So in terms of these parameters, it found all these were really impressive. Then when it comes to the performance parameters, I just compared these two aligners based on these metrics, and from this table you can figure that CellRanger is outperformed in across the board. And then moving on to the challenges I encountered. So from our benchmarking, several key insights emerged. The resource planning, it was critical. Memory and CPU usage varied significantly depending on the aligner and data size. Secondly, interpreting the quality control metrics required understanding the difference between raw and filtered data. This step often confused from a beginner point of view. Then each aligner produced unique output formats. So tool specific knowledge, it was critical to extract the comparable metrics. And finally, effective file management became essential. Since the large single cell data can be easily consume terabytes of storage. Then moving on to the key learning points. Again, as I mentioned, the resource planning is critical. Then the quality control interpretation was also essential. Tool specific knowledge and file management was also somewhat intriguing. Then let me conclude this presentation. So what we have achieved through this pilot study is we could successfully generated the count matrices using both CellRanger and SimpleLeaf. STARSolo couldn't run successfully. We extracted key metrics for a multi-aligner comparison study. CellRanger slightly outperformed SimpleLeaf on this particular inflammatory prostate mouse model. And through this process, we gain valuable hands-on experience with bioinformatics automation workflows. So that we have documented to help others avoid similar bottlenecks. So yeah, so our key takeaway is that with proper guidance, including clearer documentation, especially the configuration file and the decision framework, can become a highly accessible and powerful tool for single cell transcriptomics using Nextflow. So yeah, thank you for your attention. Before concluding this, I just want to acknowledge my mentor, Dr. Timothy L. Ratliff, and all these collaborators and funding and the service course. So thank you for your attention, and I would be happy to take any questions. Thank you.